Okay, so I will talk about sleep, but before that, probably most of you do not know me. So I would like to introduce myself a little bit. So I said born in Japan. I'm born in Japan as a scientist because I graduated the medical school in Tsukuba. And this was my lifetime decision, the most important and difficult decision not to become a physician. And then I went directly to the PhD course, and I was very lucky to discover what's called endothelin, as Professor Eileen already mentioned. And triggered by that discovery, I was kind of recruited to University of Texas Southwestern Medical Center from the beginning as a PI. This was a very unusual thing, and I was so lucky to be there. By the way, my mentors there was these doctors Goldstein and Brown, who got Nobel Prize back in '85. They are amazing. They are still active today, and they are still keeping their lab by two. They never split from each other. That's amazing. And I was also given as an HHMI investigator position there. So I was, in that sense, nurtured in the US. So although I do not have any, even one co-authored paper with Goldstein and Brown, but I really consider them as my role model and a mentor. So I was again lucky enough to discover what's called Orexin, this new neuropeptide, which regulates sleep. And that was also made as a drug. And several important things happens. But back in 2010, I was kind of recruited back to Japan by this program, also administered by JSPS, by the way. It's called FIRST. It's a huge amount of money, about one order of magnitude larger amount of resource than what I could manage in the US. So I decided to come back to Japan. First, I was commuting literally between Dallas and Tsukuba, which was very bad, right? I mean, I'm a sleep researcher and I'm in the jet lag constantly. So, eventually I quit doing it back in 2014. It was a bittersweet experience, but I decided to close down my University of Texas lab and resign from the HHMI. And I was very lucky enough this year to get this breakthrough prize, which is famous for the amount of the award money, actually. Administered by these, Google founder and so on, as you may know. So anyway, so my institute, little institute in Tsukuba, this is actually the world's largest institute dedicated for the basic science of sleep and hibernation. We don't do anything else. This is a highly focused research institute. These people are our PIs. We have quite a few PIs by now. And under these PIs, we have a total of more than 180 people working in this institute. All of them are researching sleep and hibernation, okay? And this is perhaps the most important slide, one of the most important slides today. So this is already dated back to 2021. So the number is already outdated, okay? But we have tons of international collaborations all with countries all over the world. But most important thing is Malaysia is not here. Not yet. So this is actually my recruiting talk. Please introduce to us any young researchers, any stage, undergrad, graduate school, or even young, researchers, postdocs. Any stage is fine. If you know any motivated young people who are genuinely interested in neuroscience, please introduce him or her to us. I'll be very lucky to have her here. So this is yet another slide of, we recently got another JSPS grant called Reconnect, and many of our scientists and abroad scientists, young scientists, being exchanged throughout the world. Not yet from Malaysia. So please introduce anybody to me. So back to sleep. So sleep is so fundamental of the animal behavior that all of the vertebrates, all of the non-vertebrate species sleep. They all have a brain, okay? Even though some of them are have a little brain, they all sleep. But this recent paper shows that even jellyfish sleep. This is amazing because jellyfish does not have a brain. Okay, so the message of this paper is sleep is invented before the brain. So it's very, very fundamental, okay? So among the mammalian species, these are all mammalian species. The vertical axis is the average time of sleep per day for each species. You see all mammals sleep, first of all. But species difference in sleep time is very great, from 2 to 3 hours a day up to 20 hours a day. However, you see this tendency that phylogeny is closer, the sleep time tended to be closer, right? So this set point, so to speak, for average daily sleep time is written somewhere in their DNA, okay? But how it's set, it's complete mystery. So this is the big questions, two big questions in the field. Why do we means all animals with nervous system, why do we have to all sleep? And how wake is regulated, for example, how the daily sleep amount is set. By the way, before going there, some social considerations. This is very interesting data. Each data point represents countries. Vertical axis is objectively measured daily sleep time, average daily sleep time of nationals in individual countries. The horizontal axis is actually GDP per capita. So the right-handmost countries are economically richer, wealthier countries, okay? So you already see, general, very strong correlation between GDP capita and, daily sleep time, right? Well-slept countries tend to be richer. That's very, very, very important lesson, okay? Where's Malaysia? Malaysia is not very good among these groups, relatively low sleep, okay? I know Malaysia's GDP capita is really growing now, but you should sleep better. But the problem is, where is Japan? Here. This is really, Japan and Korea actually are the kind of abnormal data point. This is really out of the group. This is a big problem in Japan, okay? So indeed, among these industrialized countries, the loss of economical loss due to insufficient sleep is much larger in Japan than any other countries per GDP. In other words, if we, the Japanese people, sleep well, we will instantly have a 3% of growth. So why is it so? One of the reasons is, of course, sleep deprivation impair your performance. So this is a vertical axis is brain performance, the upper the better. And horizontal axis is continuous wake hours immediately before the testing. Okay, so say from 7 a.m. you wake up, 7 p.m. here, you're still not very tired. But after 24 hours, 7 a.m. next morning, without sleep, your performance get there, okay? That's understandable. But in this same Nature paper, they did this alcohol clamp experiment. So they continuously intravenously inject alcohol to maintain blood alcohol level at constant level. And after one night of sleep deprivation, the brain performance is about equivalent to 0.1% of alcohol, which is complete drunkenness, okay? So, no, I'm serious. And that was complete sleep deprivation, but not even complete deprivation. 4 hours a day sleep for 7 days, or 6 hours of day sleep for, say, 10 days. It's about equivalent of complete sleep loss one night. And the problem is, this is objective performance decline. Subjective sleepiness does not go up as fast. So that's the pitfall, okay? So, you know, even those healthy young adults who claims they are sleeping enough, after being asked, you should sleep as much as possible, as long as possible, okay? Extended sleep, day 1, day 2, day 3. They sleep in the first day nearly 10.5 hours. And they cannot sleep anymore after several days, right? So the conclusion of this paper is even healthy young individuals subjectively slept enough have an average sleep debt of about 1 hour per day. And in order to pay back that debt, it takes at least 4 days. Another important message here is humans cannot sleep more than they need. This is as much as they can sleep after really paying off the sleep debt. So, you know, just one slide, representative slide for health aspect. This is just a risk for atrial fibrillation. This is, you know, potentially dangerous arrhythmia in the heart. That risk is greatly enhanced by insufficient sleep in this case. I mean, this type of epidemiological study, there are tons of studies these days. I mean, it's very, very strong medical evidence that insufficient sleep or bad sleep, irregular sleep, results in higher risks of various common diseases, including metabolic syndromes, mental problems, including depression and dementia, cancer, etc. You name common diseases, all risk is enhanced by bad sleep. Okay, so the reason we went to sleep research nearly 20 years ago was accident. We did not plan to go there. So at that time, we happened to discover this new neuropeptide called Orexin in the brain, in the lateral, what's called hypothalamus. Initially, we did not know what they do, but our mouse experiments clearly show that loss of orexin results in the disease called narcolepsy. So then, Emmanuel Mignot, my co-awardee of the Breakthrough Prize this year, discovered that in human narcolepsy patients, orexin neurons are lost very specifically. In the same region of the brain, other kinds of neurons are completely intact. They lose orexin neuron very specifically. And this is an example of orexin deficient human being. This is a patient with narcolepsy. So she is actually smiling, joking around with her mother or somebody, and suddenly she loses muscle tone bilaterally and crumbles down. So this is called a cataplexy attack. She is getting into this REM atonia, like REM sleep-like state. So again, she's joking around something with her boyfriend probably, or husband, and she went there. And he knows she will lose muscle tone. So this is very strange phenomenon. It's peculiar to narcolepsy. When we laugh, you lose muscle tone. You went suddenly into REM-like state. Anyway, so this is the results of loss of orexin. So that means, and the patients are of course sleepy all the time during the day. So that that and many, many other studies establish that orexin is the boss, master regulator of wakefulness in our brain, okay? So if you block the action of orexin, you will become sleepy. So that's already applied as a clinical medicine. These orexin receptor antagonists are now in clinical use in Japan, Australia, US, Europe, etc. And that's supposedly very, very good sleep-inducing drug. And we also study orexin agonist, which by definition should be a mechanistic treatment for orexin deficiency syndrome, which is narcolepsy. So this is, for example, our structure of our compound, which fits to the ligand binding pocket of the receptor molecule. And giving these drug in mouse, the orexin knockout mice, prevent these narcoleptic attacks. So it at least works in mice. So in this regard, Japan's Takeda is now forefront. They have their compound in phase 2B clinical trials. So, those patients will be certainly hugely benefited by this agonist once it's clinically available. So back to orexin antagonism, you may wonder, there are many sleep-inducing drugs already in the market. Why do we need another one? The reason is, the fact is that all of the currently available traditional sleep-inducing medicine is all GABA-A agonist. And they have these problems, and orexin antagonism does not have these problems, especially this lower risk of cognitive dysfunction. That's very, very important. And lack of dependency. You can stop anytime. That's very important. And talking about dementia, this is not our work. This is from WashU in Missouri. Very recent paper, just 3 weeks ago, they published this paper. This was an amazing paper. This is an acute study in very small number of patients, but once you get orexin antagonist, on that same night, the CSF levels of those substances like amyloid beta and phosphorylated tau, which is a causative agent for the Alzheimer's disease, they decrease, okay? So again, this is small acute study, but if this type of thing continues over long term, maybe orexin antagonism might be a preventive medicine for Alzheimer's disease. So back to orexin. So orexin is, like I said, the master regulator for the wakefulness, wake-inducing system. On the other hand, another lab, Oishi Lazarus lab in our institute, discovered that these neurons are sleep-inducing neurons. So their study, coming from the study of what's called adenosine. Adenosine is a brain metabolite which is known to be a very strong somnogen. Adenosine makes you sleepy. And actually caffeine, okay, caffeine is a famous adenosine receptor antagonist. That's how caffeine induces wakefulness, okay? By the way, I'm really enjoying Malaysian coffee. It's excellent. So what they discovered then is where specifically in the brain adenosine and caffeine works to induce sleepiness or wakefulness. Their answer was very unexpected. This structure called nucleus accumbens, which is traditionally linked to, say, drug addiction and reward and motivation and so on, never thought to be involved in sleep-wake regulation. That structure have these receptors, and these neurons strongly induce wakefulness. By the way, when you are very motivated or highly excited, the dopamine, another regulator, is really released here, and that inhibit these neurons to make wakefulness. So that's a very interesting study. Anyway, my point is, this is one example of strongly sleep-inducing group of neurons. So current consensus is that throughout the brain, there are different groups and networks of neurons which promote wakefulness. Another groups of neurons promote sleep. And at the neural network level, they compete with each other. They are inhibiting with each other to make this flip-flop like seesaw-like metastable switch machinery. Okay, so that much is very rigorously studied in the last 10 years or so, and this much is established. Okay, but the problem or deeper question is, okay, there is this switch between sleep and wakefulness in our brain, but what's the finger which pushes the switch? That's deeper problem. And what is this sleep, what's called sleep pressure? If you are awake for a long time, you will be sleepier and sleepier. Why is it so? Nobody can answer that simple question. That's called the homeostatic process of sleep regulation. You will have this feedback mechanism to recover from sleep deprivation, right? So this is a Japanese bamboo thing you see in Japanese gardens. So bamboo tube is upwards and water is trickling in. So that's in wakefulness, okay, wake state. And water level will goes up gradually. By the way, orexin is depicted as a nightingale to stabilize wakefulness. And water becomes at certain level, then suddenly this bamboo tube flips. That's the neural network-based switching machinery, as I said, and water flows away. In the humans, it takes 7 or 8 hours to flow away completely, and then flips back. So this cartoon very nicely depicts the sleep-wake cycle, conceptually, okay? But nobody knows what is the actual, what's depicted by the water in the brain. Nobody knows what's depicted as a bamboo tube in the brain, which is an accumulator or counter or integrator, or how the information that water filled up the tube is transmitted to these switching mechanisms. These are really deep, mysteries, I would say. So our approach to this mystery is depicted by this anime. Oh, maybe the sound doesn't work, maybe. Okay, that's fine. Let's skip it anyway. Okay, so what we did was what's called the forward genetic approach, which is usually taken by fly biologists and yeast and E. coli and so on. But we did it in the mouse, which is very costly and labor intensive, but fortunately, this first program allowed us to do this. So what we do is to introduce random genetic mutation in this male mouse and screen this heterozygous offspring, literally thousands of these mice for sleep abnormality. And whatever sleep abnormality we detect, then in the next backcross generation, we do a classical genetics to determine the causal mutation, which causes this sleep abnormality. So that's said easy, but actually doing it is very labor intensive. So our mouse room have this type of facility where these are individual mouse cages and mice are sleeping here very comfortably. The job of our technician is to make them very comfortable, okay, all of them. That's very important. And anyway, so within this one room, we can parallelly measure the sleep-wake of mice, more than 100 mice parallelly with a EEG, EMG recording. So years ago, we could find what we call a literally sleepy mutant pedigree, which these sleepy mice sleeps like 5 hours more than normal mice. In the example of humans, say average, your average sleep time is 7 hours, then this individual sleeps 12 hours a day. And still they are sleepy, okay? So we established, in a sense, a entirely new sleepy, model of sleepiness. So we thought that, traditional way of making sleepy animal is sleep deprivation, right? Sleep deprive the mice and they are sleepier. But the problem is you cannot dissociate the effect of stress. Sleep deprivation is always very much of the stress for the animal. By the way, it is stress for the graduate students who's doing the experiments. And also, you cannot easily dissociate the effect of wake state versus effect of increased sleep need. So another, triple ISPI group, Jinhua Liu's group, did this experiment. So like I said, the top of that is a conclusion. So sleep deprivation, they did not sleep, so they are sleepy. Sleepy mice sleeps more than normal mice, but still they are sleepier. So we cross-compare these conditions and take a common denominator here that should be correlated with sleep need. And to make a long story short, we found that, oh, by the way, the original mutation in the sleepy pedigree was in the protein kinase gene, the gene giving the phosphorylation phosphate group to the certain specific location of certain specific proteins. It's called kinase, and that's mutated in the original sleepy mice. So we looked for phosphorylation difference in the brain. And to make a long story short, at that time our conclusion was certain subsets of proteins which is known to be localized in synapses, those location where the two neurons meet with each other and transmit signals with each other. Those protein, the level of phosphorylation seems to be one substrate for sleepiness, okay? So, but of course, there are lots of other hardcore questions remaining. For example, this sleepy mutation, which is, of course, original mutant mice have this mutation in the germline everywhere in the body. Where in the brain this sleepy mutation works? Our current answer is very interesting. So the same molecular pathway regulates sleep depth. This is a index of sleep depth. Sleep depth and the sleep amount, sleep time, in different locations of the brain. So in the hypothalamus, this SIK pathway regulates sleep amount. In the cortex, the same molecular pathway regulates sleep depth. And within the hypothalamus, this is unpublished work by one of my graduate students. Within this type of region, the anterior medial part of the hypothalamus, it seems to be regulating sleep amount. So this is ongoing work. Another obvious question is, okay, this sleepy gene encodes for protein kinase. If that's the case, then what is the upstream regulator? What is the target molecule phosphorylated by this kinase, etc.? Molecular pathways. So to make a hugely long story short, recently we could publish this type of molecular pathway, so-called, seems to be representing and regulating sleep amount and sleep depth in the brain, okay? So we are getting there, but it's still a long, long story ahead of us. By the way, so this is to be continued. This is not a Photoshop trick. When I was living in Texas, 20 years ago, I was working on this This is my real Honda Odyssey minivan. Oh, with this license plate. So every time, at that time, the expressway, the fee was collected by the person, not the card. And every time I goes to those, expressway booths, they ask, what the heck is Orexin? I don't answer because it will cause a traffic jam. Okay, so this is a little acknowledgement of the teams of collaborators and importantly, funding agencies. JSPS, please. Anyway, so last several minutes, I completely switch gear to more of the real world implementation, application type of study we are doing, okay? This is interesting, very old, but interesting data. Everybody seems to know what's called insomnia. Insomnia is a condition where you cannot sleep even though you try to sleep, right? That's the insomnia. However, this is interesting data. This horizontal axis is actual sleep time per night, measured objectively by EEG. Vertical axis is the patient's complaint, subjective sleep time. So for example, this particular patient, oh, by the way, these white dots are normal people, healthy people. They are on this line, which means they can assess their sleep time correctly, right? However, this insomniac patient, she complains that she can sleep 3 hours a night. But objectively, she is sleeping 8 hours. This is called sleep misperception, sleep state misperception, okay? They cannot measure their own sleep correctly. She's not lying or anything. She really feels that she can sleep only 3 hours a day. But these patients, for example, also complain that she cannot sleep more than 3 hours, but she is actually sleeping only 4 hours. So this person has objective insomnia, okay? Everybody probably here agrees that these patients and these patients should not be treated the same way, right? It's a different disorder, I would say, objective versus subjective insomnia. But in reality, today's sleep medicine anywhere in the world treat these patients completely identically, simply because they do not objectively measure sleep, period, okay? So why? Because medical grade measurement of sleep requires what's called polysomnography or PSG, in which you have to sleep in this spaghetti state, usually in the hospital. So you cannot, PSG cannot measure your native, natural sleep state. It's very artificial. I mean, many people cannot ever sleep well like this. And all over the world, but for example, in Japan, there is only like 300 facilities and 1,000 beds all over Japan who can do this. So capacity is very limited. Okay, all over Japan, only like 100,000 tests per year was conducted, but there should be more than 10 million, sleep disordered patients latently, right? Potentially. So 2 orders of magnitude deficient. So we thought that maybe, like 60 or 70 years ago, multiple Japanese companies invented this very easy blood pressure measuring device, and everybody uses it, and that really changed high blood pressure medicine. We maybe be able to devise EEG-based sleep measurement at home by themselves. So we developed recently what's called insomnograph, in which you just place a sticker-like thing in your forehead and the back of the ear. That's all. You can measure your own sleep EEG. And of course, these simple EEG device, all of these kind of, characteristic waves for each sleep states are reflected in the data. And these data are collected and immediately sent to the cloud. This device by itself is an IoT device. And in the deep learning type of scheme in the cloud, it automatically deduces sleep states. And currently, we achieve very, very high accuracy. Some of you may say, Fitbit, Apple Watch, those wearable devices can measure sleep pretty accurately. That's half true. Yes, this is Fitbit example. The gold standard is PSG. This is epoch by epoch. One night is divided by 30-second epochs, and epoch by epoch accuracy. To be honest, in our hand, Fitbit is more accurate than we thought. So it's usable. But of course, our device is EEG based, so it's much more accurate. Anyway, so this is one example of kind of field study we did recently, very small observational study. So in checkup clinic in Tokyo, we recruited 100 people, and we did a very large set of standard health indexes, blood tests and so on. And subjective sleep quality, we used what's called Athens Insomnia Scale. It measures, this is a questionnaire which measures your subjective sleep quality and objective sleep quality by EEG. And we did unsupervised computer clustering based on this objective sleep quality, okay, into 3 clusters. So this is a conclusion. So these 3 bars, this is Athens less than 6, which means subjectively good sleeper. This is bad sleeper, subjectively, these 3. And within these groups, objective sleep quality, good, middle, bad, good, middle, bad. And interestingly, the vertical axis is in, for example, systolic blood pressure, simply, very simply, maximum blood pressure. And objective sleep quality is very important indicator or predictor for blood pressure in this case. Subjective sleep quality does not matter, okay? So this is one example of field use for this type of in-home EEG device. So this is my last slide. Sorry for Japanese slide because this is from Japan. This is Japan's data. So this is Japanese gap, unfortunately, Japanese gaps of biological and healthy life expectation. In other words, lifespan versus health span. In males, it's average 9 years. In females, 12 years. And it's not shrinking, right? Life expectancy is constantly elongated, longer life. But unfortunately, this gap is not shrinking in Japan yet. So this is the theme of this symposium, right? I mean, and we believe, like I said, healthy sleep, sound sleep is the key to shrinking this gap and having a productive aging. Okay, so that's all, I guess. And this is our building, and I repeat, please introduce to us any motivated young researchers who are interested in neuroscience. Thank you so much for your attention.
Thank you. Thank you. Thank you, Professor.